Hello everyone. So today we will perform protein modeling, that is lab number 4. And protein modeling means converting DNA or mRNA sequence into protein, then conversion of that sequence into amino acid, which is called translation. So the first step is transcription, and the second step is translation. So after translation, that means converting the mRNA sequence into amino acid sequence, we go for making the model of the protein. So as we cannot see the real structure of the protein, in case of bioinformatics, we predict the model of the protein. So today I will show you the available tools for getting the DNA sequence or exons that are actually the expressible part of the DNA, and using that exon to translate the sequence into amino acid. Then we will use that amino acid sequence to perform the protein modeling. We have the tools for secondary structure modeling. We have the tools for homology modeling. We will use Swiss model for that. Then we have a couple of questions to answer. And also we will use iTaser. Yeah, so these are the tools that we will use today to make a protein model. So firstly, I'm going by the procedure. We need to open a DNA sequence file. Here we will use Ensembl. So I'm just copying the word Ensembl. I'm opening a new tab. And just pasting the word Ensembl. Yeah, so this is the tool to get a DNA sequence. We have several other tools, but today I'm using Ensembl. So here in the first search box, you have to select the organism or species, selecting human. And here, according to the protocol, you are supposed to search for human HR gene, because it is a very common gene and we have performed it before for lots of times. I'm just changing the name of the gene so that you can experience something new. So I think I will get a very common gene, BRCA2. So you can directly go to this suggestion, or you can also type capital BRCA2, which is a gene found in human and many other species as well. So today we will work with human gene BRCA2. It's actually breast cancer gene. And I will click on the first option. Now, here, after tapping BRCA2, I got the page here. Now I will go to this toolbar. There is an option called sequence. I will click on the sequence to get the sequence of human BRCA2 gene. So this is the sequence of human BRCA2. It's a huge sequence, as you can see. So these are exons regions, those are highlighted here. These are exons. Exons means the expressible part of the DNA. Expressible part of the DNA means the part of the DNA that can be translated into protein, that has expression capacity. Okay, so out of all the sequence, out of all the exons, I will take this region. It's pretty small, so it will not take time to make a protein out of it. So I'm copying the sequence for now. And so the next step is to make a text file in your system for the nucleotide sequence. So what I can do is I just can make a sequence, make a sequence file. I can just use Microsoft Word. I can use Notepad or text documents. Here I'm pasting the sequence. I'm saving the file as BRCA. Yeah, okay. Now the next step is this question: which exon you are going to use to model your protein? So because I have copied the sequence, pasting it here. So this is the sequence that we will use to model the protein. All right, I'm saving the information. Now, protein sequence file. So this is the DNA segment that we got for BRCA2 gene. It's a part of the gene. Now, we will use the protein sequence file to translate this sequence, this DNA sequence. So we don't need to transcribe this DNA sequence into mRNA. We can directly translate this protein, this DNA sequence to amino acid sequence. So open XPressy translate and copying XPressy translate, or just can type XPressy translate in a new tab. So this is the translation tool. I'm getting into this tool. So this is the view of the XPressy translate tool homepage. And there is a box where you can put the DNA that you have copied before from here. Copy and paste in the XPressy translate toolbox. I'm putting backspace to minimize the size here. Now, just need to click on translate. And if I go, so if I scroll it down a bit, so these red portions are for open reading frame. Select your initiator. Out of these three out of these three sequence. Actually, this is 5 to 3 frame 1, 5 to 3 frame 2, 5 to 3 frame 3, and the rest of the sequences, these are the opposite of these chunks, these sequences, because these are from 3 to 5. This is from opposite direction. So I will take, because L is also read here, so it is also an open reading frame. I will take this sequence from 5 to 3 frame 3, copy the sequence, and then paste it below the question that which ORF I'm going to use. So this is the ORF that I'm going to use. Use for what? Use for making the secondary structure of the protein. So to make a secondary structure of the protein in section 3, we have to open this server, PSIPRED server. I will copy the name and I will open a new tab and paste there. So PSIPRED, this is a workbench. It is for making the secondary structure of the protein, not the tertiary structure. It is for the secondary structure prediction. So if I click on the link, this is the page where we can make the secondary structure of our query sequence. So the protein sequence that I have taken from explicit translate tool was this, the third one from 5 to 3 frame 3, and I'm pasting the sequence here in the box. I'm pasting the sequence here, copy, pasting it. Then you can give a job name, BRC2, okay. Email is optional. If you give them your email, they will send you the image in your email. Image means the secondary protein structure image. So I'm submitting the work. So it will take some time to be loaded. So meanwhile, we will go to the next section. So they were, it is described as like entering password and short identifier. We don't need to do that. Before we used to give an identifier to, or we were given an identifier by PSIPRED, but nowadays it is not a requirement. So this question need not to be answered. I need to answer this. Okay. So the next is homology modeling structure. This is for 3D structure prediction of the protein. For that, we will use Swiss model. Opening a new tab. Swiss model. Okay, this is the home page for Swiss model. This page is used for, tertiary structure prediction of the protein. And getting back to the PSIPRED workbench, the secondary structure prediction of the protein for BRCA2 gene is done. This is the secondary protein structure. So you can get the PNG from here, and you can see the highlighted portion. These highlighted portion are for, the pink ones are for helix. So that means they are helical structure. So what we will do is we will take a screenshot. I use snapping tool usually. So I take screenshot with snapping tool, copy the image and paste it to the manual. Just below the question that we didn't answer about the identifier. We don't need the identifier anymore. So I am placing the image that I got, the secondary image, secondary protein structure image that I got from PSIP-RED. And then we went inside the Swiss model, and then we need to press start modeling, and then we can upload a file or paste the sequence that we get from the protein translation tool to build a model. So we have to give a title to the job and email address as well, so that they can send you the results in your email. Now, getting back to the Swiss model. Yes, so start modeling. So this is a blank box here to paste the amino acid sequence. So amino acid sequence we got from Expressly Translate. That was a frame 3. So, okay, I am copying the sequence again and pasting it to the Swiss model box. I'm giving the project title the same, BRCA2. It's all right. Email is optional. If you give your email address, they will send the results of your search in your email. So I'm giving an email address. I'm giving my institutional email. Now, build model. It's being loaded. Let's check the worksheet. So, image for proteins. So we need to answer these questions from the loaded page. Comparison with non-redundant set of PDB structure. PDB means protein data bank structure. So they have some comparison of their results with PDB structures. I don't need to read the comparison, and you cannot interpret the comparison right now. Just put the image that you get from Swiss model, which is still being loaded. It's a bit slow, so we need to wait a bit. Okay, in the meantime, we can also go to the next section to see what we have, what other tools we have for AB initio modeling of the protein. We have iTaser server. So I will open iTaser. Okay, the job in Swiss model is done. So we can open iTaser later. This is the protein structure that we got from our sequence. Okay. In Swiss model, they have made the model out of our sequence. So the question was comparison with non-redundant set of PDB structures. So So if we click on the model, there are three sections here: summary, template, and models. If we click on the model, they show us the comparison with PDB structure here. This is the local estimate one. Here, this is the comparison with non-redundant set of PDB structures. So we can take a screenshot with snapping tool. You also can save this image as PNG. So I'm copying the image and I am pasting it here, saving the information. Okay, so this is the comparison with non-redundant set of PDB structures. In local quality estimate, getting back to the page, closing the image. So this is the local quality estimate of your protein. So I will take a screenshot of this local quality estimate. Copy and paste to our manual below local quality estimate. Okay, saving the information. Now, in section C, they're asking for 3D structure of the protein that we got in Swiss model. So it was the 3D structure of the protein that we got in Swiss model. Using snapping tool, I'm taking the screenshot, copying the image and pasting it just below the section C. So this is our 3D structure built by Swiss model. Here, question number 2, go to the template section. How many templates you have for the protein of interest, and how many templates have ligands. Copy and paste the image you found for the template 2B5X1 and 2B5Y1A. All right. So getting back to the thing, they are asking for templates, that how many templates it has. How many templates? It is shown as 16. So we'll write the answer as 16. How many templates? Okay. Answer 16. And then how many of the templates have ligands? We also have ligand information. These are ligands. So you can see that these are the templates, and you go to the ligand section and you can find out the number of ligands that these templates have. Out of 16, there might be ligands for all the templates, or there might be ligands for few of them. You need to find it out, okay, out of this information. Now, so this question you need to answer, this one. How many of, this is for you, all of you. All right. So copy and paste the template. So these templates were for gene HR. This cannot be available for BRCA2. So we will take for BRCA2, We'll take the first one. Let's wait for it to be loaded. All right, so this was the first template. I have been in the template result page in the template section. So I clicked on the first accession number. It is a first template, which is 5WAU.1.G. So this is the result I got for the template. We'll just take a screenshot for the result. Copy and paste it to my manual. Yeah, saving the information. So this is not the template. For us, the template is this one. So I'm copying the number 5WAU.1. For template. For the template, this one. All right, saving the information. Now they ask for the image, so I have put an image here. Then how many templates? There were 16. So this is the answer, 16. Now go to the quaternary structure, make a combined image with all the templates. So I'm getting back to the Swiss model again. Going to the quaternary structure. In the first page. So as you can see here in the image section, there is no template selected. That's why there is no image. So I'll select all the templates here and you will see a combined image, because whenever I click on the image, it will show you a combined image. So this one. I'm selecting all the templates. So the image is increasing with the template numbers. So these are all the possible templates available for the protein. And if you just press hold the touchpad and move a bit, it will show you the protein moving. Also there are option for start rotation, so it will rotate. If you want to see the rotation in the full image, it will rotate for you. You can pause it. Yeah. So if you want to take the screenshot like this to answer our question, you can. You can use screenshot with all the possible templates for the protein. This is the combined image. So copy and paste it to the manual just below the question number 3. Okay. Then question number 4 is go to the sequence similarity, copy and paste your result image. So I'm getting back to the page. Go to the sequence similarity option. So they asked us to go to the sequence similarity. Okay. Sequence similarity means how many genes or structures are there, like proteins are there, who are sharing the same sequence with us. So this is a graphical dot plot presentation. I'll take a screenshot for template number 5 WAU.1.G. Copy and paste it below the question number 4. Save. Then moving to iTaser for AB initio modeling. Before that, I want to show you something. In Swiss model, we opened a page for template number 5 WAU.1. Here also you can select a particular ligand or just place your cursor to a particular ligand. So in the image, you can see that portion will be highlighted or that portion will be a bit apparent. You can see. See, the whole protein is being a bit blurred and a particular portion is being apparent. If you want to see the structure in detail, you can always start rotation and the structure will move for you. And also you have detach option. So in the detach option, they will give you a separate, in a separate window, they will give you the option to watch the structure separately, like being detached from the page. So we need to wait for a bit for the structure to come. Yes, so yes, so this is a structure that was being detached from the website and they have made it for you. Yeah. You can move through the whole protein structure. Wherever you want to go, you can see the ligands, you can see the helical structures. Yeah, you can move forward, move backward as well. And also you can play the rotation to start rotation. Then they will show you the whole image and the structure of the protein. And it will move to every corner. You can see helical structure here. We can see the ligands as well here. All right. Now, I'm opening iTaser. iTaser. This is also for protein structure and function prediction. But in iTaser, it is important for the people to register in iTaser first. So this is provided by University of Michigan, and yeah, so let's get back to the manual. So we need to enter our email address in iTaser. We need to get registered and have password. Then we need to enter a protein ID that is given by the iTaser to us. Then we need to submit the job. So what is ab initio modeling? That is for you to answer. I will not answer it for you. And here question number 2 is how many proteins are predicted by iTaser to date? Means how many proteins are predicted by iTaser to date. I think this information you can have just below the logo. The server completed predictions for 643651 proteins submitted by 15,000. It's not 15,000. It's actually 155351. So yeah, that many users from 158 countries, they have submitted the job here in iTaser to make structures for their protein. Yeah, so how many proteins? This is the number of the protein, 643651. I'm copying the number and pasting below the question number 2. All right, saving the information. Now, so the last question is how many predicted models you have found for your amino acid sequence. Add your model in the answer sheet. For that, we need to submit the job first and we need to register in iTaser. So I'm going back to iTaser homepage. To register in iTaser, you need to put your email address. I have put my institutional email address. And after I put my email address, I put a password. So my password are saved, and I put an ID BRCA2. For you to get registered in iTaser, you will give your institutional email address here, and then you will not put any password. You will give ID to your job, and you try to run iTaser. And of course, before running iTaser, you need to put the protein sequence that you got in XpressC2, this one, that is the amino acid sequence. You need to put it here in iTaser, just like as you did before. You put the sequence in Swiss modeling, you put the sequence in PSIPRAD. Okay, so just like that, you need to put the same amino acid sequence to iTaser box. When you do this task without putting your password, they will tell you to put a password. So for that, if you cannot give a password because you do not have a password from iTaser, iTaser will email you with the password, and they will ask you to put the password for the registration. So you need to wait for the email from iTaser. Okay. So iTaser, after you are done with the registration, iTaser will give you a notification like this. After you're done with the registration and adding your job to the site, they will give you a job ID that you need to remember. You can take a screenshot of the job ID, because if they do not contact with you with the protein structures, you have to email them in the email address. Okay. So I am running iTaser. So I already have submitted one job. So that one job is pending. So you cannot submit more than one job at a time. Because it's a very comprehensive job server. They have a lots of job in one day to perform. So I have to wait for my result. They will email the results with the protein image, I think within 24 hours. And in better cases, they can email us within 6 hours or even 2 hours. So if 24 hours is passed and you do not get any email with the protein structure result, you can email them. They will provide you with this email address after you submit your job, and they will ask you to email them if you do not get your result within the time. All right, so yeah. Yes, so yeah, that is protein modeling lab. I hope you could perform it better than me. But if you face any problem, feel free to email me, especially regarding the registration in iTaser. It's a free registration, so I think you won't face any problem. And thank you for being with me.